Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12, the chapter Molecular Basis of Inheritance, Part 14, Human Genome Project. The preceding section you have learned that the sequence of bases in the DNA determines the genetic information to give organisms. So when you take as any one particular organism, it contains as in what the genetic information is present, right? In this genetic information to get arranged as in how the total genetic material, already we know that is what it is a part of the DNA, right? So the DNA to get arranged as in a one sequence manner. Next, in other words, the genetic makeup of any organism or individual alive in the DNA sequence. So in this total DNA to get arranged on the completely in the sequence process. If two individuals differ, their their DNA sequence also is different. Here, the DNA, for example, it is a one strand, and here as in another strand is there, right? So the DNA sequence completely they differ for as in organism to organism and DNA to DNA. See, at least some places. So, for example, here adenine is present, here thymine. Here guanine is present, here cytosine. Like they're complete the some sequence, the changes will be there takes place. This accomplishment lead the quest of in the finding out the complete the DNA sequence of human genome. So you can easily to get find the enter the DNA sequence based on that easily to get identify that belongs to as an A which organism DNA. Okay, and with the establishment of the genetic engineering techniques where was impossible and isolated and proven the price of the DNA, the availability of the simple fast techniques the determining the DNA sequence very their ambitious project of in sequencing the human genome it launched the in year 1990. So yearly the how to they easy to get identify the your human genome DNAs, they can easily to identify the based on they are launched as in one the project will be the launched in 1990, that is what Human Genome Project. That is what Human Genome Project. It was called as a mega project. So this Human Genome Project is what it is in a completely mega project. You can the imagine the magnitude and the requirements for the project. It be the simple the defined aims of the project follows. So it is a large project. It is completely get follow as in some sequences one by one. Let's see here. The genome is said to have the approximately in 3 into 10 to the power of 9 base space. Already we know that any the complete the DNA sequence how to get calculated in that 3 into 10 to the power of 9 base pairs. In the cost of the sequencing, the required that completely the US dollars in a 3 US dollars base pairs, the estimated cost in the beginning. The first they are $3 they are estimated. In the total, the completely estimated cost of the project would be the approximately in 9 US dollars. So now the estimated as in that completely to develop as in that human genome project, they are completely $9 US dollars. For the further, if they obtain the sequence where to obtain the stored any type of if the any type the typed from in books. So the completely what kind of the genetic sequence is present, they can easily their typed information and each page of the book they contain as in 1,000 letters and each book contain as 1,000 pages. So in this total the human genome book they are completely how many pages? The 1,000 pages, and within the 1,000 pages they completely around as any they each contain as 1,000 letters also they are present. Then 3,300 and such books would be they are required for the storage of information the DNA sequence from as in a single cell. So here around as in 3,300 and completely their books will be there required for that in a storage of in the DNA sequence of information, okay, for the cell. Next, enormous amount of the data they are expected to be the generating is also the necessity is and the use of any high speed computational the devices for the storage and their completely retrieval and analysis. So this total information based on that, the speed communication based on, we can easily to get to develop as in a that human genome project. Okay, and the human genome project was in closely associated with as in a rapid development of new era. The biology is called as in bioinformatics. So in this human genome project, to get help for as in to get the new things they can easily get identified. Okay, so nowadays it is we are called as what in bioinformatics. Okay, what are the goals of human genome project? Let's we discuss here goals of human genome project. Here some important goals of human genome project was followed. What kind of their goals as present? The first one, identify all the approximately the 20,000 to 25,000 genes in the human DNA. So here approximately how many DNA is required? Around as in 20,000 to 25,000. And second one, here determine the sequence and the 3 billion chemical bases, the chemical base pairs make up of the human DNA. So here when human DNA is present, how many that chemical bases were required? Around as in 3 billions the sequence of any chemical base pairs are to be required. Next, the storage this information is database. The total information where to get stored, that completely get stored as in a database. Next, the provide as a tools for as in data analysis. So here what kind of the data they get stored, the total the human genome, that to say total base pairs we get how to get stored with help of in a their tools of data analysis. Okay, next, the transfer related technologies to be their sectors and such as any their industries. So in this total the related technologies we can easily to get used as in the sectors and their industries in this total human genome project we are using. Address and ethical, legal and the social issues and that maybe they arise from the as a project. So this project we get help to as a the number of ways like ethical, legal and social issues. This everything they're controlled in this human genome project. Okay, and then after here the human genome project was 13 years project that coordinated by as in US Department of the Energy and the National Institute of Health. So in this human genome project, it is around as a 13 years project. It is organized by, means coordinated by the department is what? US Department of Energy and the National Institute of Health. So this institute to get developed as in this human genome project. Next, during the early years, the human genome project in this welcome the complete trust in the UK. So in this UK, they first developed as in this project become the major their partner. The additional contribution came from Japan, France, Germany, China and others. So in these other countries also they given as a contribution for as a development of the human genome project. Okay, and then the project was in completed 2003. So when they get start, it is completely they start with as an A. They completely in 19. Here they given clearly at the start it around as in 1990. Here complete as where it is complete as an A that 2003. The knowledge about the effect on the DNA variations and among the individuals can be lead to their completely revolutionary new the ways the diagnose and treat, and then some days they prevent thousands of the complete disorders and effect of the human beings everything. So in this they completely get help to as what it is to get completely to get help to as any to get discovery of new diseases everything. Okay, besides the providing the clues to as in understanding the human biology. So it is mostly they get providing as information the understanding of human biology, learning about as a non-human organisms or DNA sequences can be lead to as understanding that they are in natural capabilities that can be there complete applied towards the solving the challenges healthcare, agriculture and energy production. Okay, and the environmental remediations like. So these are completely in this human genome project to get help to as the several ways. It is challenging as in everything, but it is completely to get do as a number of help to as in a human beings like. Here they are challenging where like as in healthcare, agriculture and the energy production and environmental remediations. So like this purpose also they get challenging. Next, many non-human the model organisms such as in bacteria, yeast and canero Canarohabditis elegans, the free living non-pathogenic nematodes. These all things they complete they get the number of the DNA sequence also they get easily identified. Like as you see the fruit fly Drosophila, the plant rice, and their Arabidopsis, etc. This completely is also they given as in their DNA, their sequence they get arranged. Next, what are the any methodologies? What are the methodologies? Next, the methodologies involve the two major approaches. One is the approach the focused on the identifying all genes that are expressed as in a RNA, that is expressed sequence tags. That is what expressed tagged sequence. That is first one is what? The first one is expressed sequence tags. Other took the blind approach, the simply the sequencing all that genome that is contained that all coding and non-coding sequence. So, for example, in this DNA, they having as what coding and non-coding is present. The coding will be they get separately the coding, non-coding organisms they get separate as in a individually. Okay, and later completely assigning the different religions and in a sequence the functions here that sequence annotation. So here the complete the different regions. Sorry, here the different the regions. For example, sometimes the DNA sequence here, sometimes the DNA sequence here. Like the different regions, they completely the DNA will get modified. That is called as what? That is sequence annotation. For the sequencing, the total DNA of the cell is isolated and converted into as in a random fragments and relatively the smaller size. The recall the DNA is very long polymer and they are in technical limitations and they're sequencing the very long piece of the DNA. So already we know the DNA has any very long polymer chain, they having as a very number of sequences is present. And the coiled it is in a cloned the sustainable host using the specialized the completely specialized vectors. So how do they get cloned? Which will get using as a vectors. Some vector get using to get easily to get modified to as in the number of the genes which they get produced. The cloning resulted into as an amplification of the each piece of the DNA fragment, such as in a suitable sequentially could be that the sequenced wise is. So every DNA to get completely what the sequentially to get arranged, sequentially to get arranged. The commonly used host where the bacteria and yeast, they are vectors where the called as a name the bacteria artificial chromosome. So in this total the DNA sequence how to get changing? You get changing with help of the bacteria artificial chromosome and yeast artificial chromosome. That is in a yeast artificial chromosome. These things based on we can easily get change as in that chromosome sequence. Remaining we discuss in this class. Thank you for watching. Please share and subscribe. Thank you.